students, welcome back to Applied Environmental Microbiology. Today we will continue our conversation on biosensors, and I will tell you about different kinds of biosensors that we have not talked about earlier, and then we will look into applications of biosensors, where they are applied, how they are important, what is their relevance and how we can use them. So, let us get started. So, the kinds of biosensors would be calorimetric biosensors, potentiometric, amperometric, optical, piezoelectrical. These are the ones we are going to briefly cover today, and just by looking at their names you can get an idea of what they are measuring. For example, piezoelectrical biosensors, let us start from the bottom of the list. Piezoelectrical materials convert mechanical action into electrical, electrical action into mechanical. So, piezoelectrical devices, in case of biosensors, they use gold to detect the angle at which electron waves are emitted when substance is exposed to laser light, or crystals such as quartz which vibrate under the influence of electrical field. So, what we have here is basically you have your piezoelectrical material and you have your bioreceptors, because this is biosensor. So, we are talking about bioreceptors, and when they interact with the analyte they undergo change, and when they undergo change the mass of, there is an addition to the mass here because some analyte has been added. Other objects that are not targets, they will just pass away, they will not attach to the piezoelectric material, but if you have the analyte then it will add to the mass, and in this way the mass changes. When the mass changes the frequency would change, and by noticing the frequency of the change in frequency of this piezoelectrical material you can get an idea of how many analytes were detected, and once you get that idea you have detected and quantified your analyte. This is how piezoelectrical biosensors work. Then we have electrochemical biosensors. What they do is in electrochemical biosensors we look at movement of electron in redox reaction when a potential is applied between two electrodes. So, we have two electrodes and we have current flowing through them, we apply a potential, and as the bioreceptors they receive the analyte and they change, then the electrochemical properties would change, the conductivity would change. Then we have potentiometric biosensors, they check the change in pH. So, for example, if you have the right analyte and it is attaching to a biosensor, the pH would change. For example, you might have an enzyme, let us say this is the surface of your biosensor and here you have enzymes. Okay, these are your enzymes that are attached to the surface. So, when the analyte comes and fits in, it is quite possible that one H plus is released or one H plus proton is accepted. So, in this case whether proton is being released when analyte interacts with the bioreceptor, or if proton is being accepted from the solution, the pH would change, it will either increase or decrease. And by looking at the change in pH we can get an idea of how much analyte is present and definitely whether it is present or not. And here on the right panel I have example from our previous lecture of ion tolerant pH based sequencing. This is not biosensor, but this is another sensor that detects and enumerates the sequence of our genetic sequences, genetic material using a pH based electrode. So, it boasts that it has the smallest, very accurate, reliable pH meter. So, we can use these really small pH meters to multiplex different kinds of biosensors. Alrighty, next we have optical biosensors. Now, I wanted to give example for optical biosensors of other sequencing techniques, the third generation sequencing techniques such as Illumina sequencing, pyro sequencing especially, because when the sequencing reaction happens or when the unsequencing happens, depending on what kind of technique it is, light is emitted and then we can detect the photons, we can take pictures and then we know whether we have A, T or G or C, again not biosensor but they work on optical based measurement. Similarly, we can have optical biosensors and we will be talking about it briefly today. Then we can also have photometric for light intensity. So, basically photon output for a luminescent or fluorescent process can be detected with photomultiplier tubes or photoiodide system. Alrighty, so now this is the time for us to discuss the optical sensor. So, a very good optical sensor, and by the way this is from a paper, this panel on the left is from a paper in Science or Nature, one of the leading scientific journals, and what you have here is a light source and you have the detection of light source. So, what in this particular chip you have this tiny red balls which are actually gold nanoparticles, and then there are aptamers attached to it. Now, let us look at aptamer. What is this word aptamer? Aptamer is a polynucleotide. Basically, it is a fragment of genetic material. So, it is a short, maybe up to 30 base pairs sequence of genetic material. You can sequence them in lab, in industry, and they can have any sequence. Or they happen to perfect complement for some sequence present in the surface of bacteria or presence in some genetic material that you want to detect, you want to analyze. Or at times we have aptamers that are very specific to certain environmental contaminants, even when they have nothing to do with genetic sequence. Because you see what happens will be this aptamer will not stay as a straight line, but it will take some 3D conformation, and we can design aptamers in a way that they are perfect fit for a particular contaminant we are interested in. So, if this is your conformation for aptamer A and it fits in perfectly pesticide A, so once it does it, we can transduce this into a readable signal. So, what you have here is these red gold particles, gold nanoparticles, and then aptamers attached to it. The bacteria that happen to have proteins or happen to have sequences on their surface which is specific to the aptamer attached, they will attach here, and when they attach here, then you can shine a light source and you will see a differences in the visual properties. Why does gold nanoparticle change its optical properties? Let us look here. So, this is your center gold nanoparticle, these are your aptamers attached. Now, you add cocaine. Cocaine is the drug, the target you want to detect, and what cocaine does is it makes these nanoparticles agglomerate. So, it acts like a link between two nanoparticles here, and when this is cocaine, by the way, the one I am highlighting in red. So, when they attach, the nanoparticle, the effective size of this conglomeration of nanoparticles of gold increases, and this is the beauty of gold, that as its diameter changes, nano gold, its color changes. So, if you take your gold nanoparticle, you add blank water with no cocaine, and then you add one with 1 millimolar cocaine, you can visually see the difference in the color, and this is why gold nanoparticles are very important. So, let us look at more qualities of gold. Gold is very much in demand metal for people making nano biosensors, because as gold agglomeration changes, as its diameter, nano gold particles diameter changes, or even its structure changes, the color changes. For example, if these are all except for the red one here, all of these are gold spheres. So, when gold is 40 nanometer, it gives this color, when it is 80 this color, 100 this, 50 nanometer this, 100 nanometer this, not depending upon what their structure is, by the way, and what their agglomeration style is, and if it is nanoprism at 100 nanometer, it gives red color. So, if I have gold starting here, but after meeting the analyte it changes and comes here, I will see a change in color, and then I know already my target is present. So, this is your gold again, and notice here, as these are nanorods, as aspect ratio changes, the color changes. So, if you have some target that will change the aspect ratio of nanorods, then you can use this calibration to find out semi-quantitatively or quantitatively the presence of your target and how much is present. And then if you have nanoshells like we had in this particular slide, nanoshells of gold. So, in this case what you can do is if you have target that when it will attach to your gold, it will change the shell thickness of nano gold, then you can use this, because here it is colorless, and as shell thickness increases you have different colors. Then nano cages, this is if you have nano cages, then it depends on the percentage of gold. So, we can use any of these properties of gold to make sure that we have reliable detection technique. So, and this is used a lot for making biosensors, because what we can do is we can take gold and we can attach aptamers to it. and these aptamers will be are designed in a way that they are specific to a particular contaminant, an analyte A. Now, when this analyte will attach to this aptamer, it makes them agglomerate, and when they agglomerate, their qualities change, and then this can be detected visually. This can also be detected using microscopies like SIRS. This perhaps is a SIRS, surface enhanced Raman spectroscopy. Alrighty, now let us look at calorimetric biosensor. If the enzyme catalyzed reaction is exothermic, you can measure the heat. For example, we know that what enzymes do is they lower the energy activation barrier for any chemical reaction, and that is how they catalyze the reaction. And so, the reaction that will never take place in room temperature happen at room temperature in living beings, in living cells. So, but if there is an enzyme catalyzed reaction that is exothermic, it creates heat. What you can do is you can use thermistors. Thermistors may be used to measure the difference in resistance between reactant and product, and hence the analyte concentration. Then we have electrochemical DNA biosensors, which example I was giving you, the aptamer based biosensors. The steps involved in electrochemical DNA hybridization biosensor involve formation of DNA recognition layer. So, I need to make a layer that will recognize the DNA, and then I need to allow the actual hybridization event to happen. Let us say this is gold nanoparticle, the actual hybridization to happen, and post this I need to allow to transform this happening into an electronic or electrical signal. And then once I have got the signal, then I can read it. So, this is exactly how it works with gold nanoparticle based sensors. For example, here you have a recognition element attached to your gold nanoparticle, and then you have the analyte that attaches like lock and key with your aptamer, and then they agglomerate and they change the this is how the agglomeration happens. They change the optical properties which you can read. Your camera can read this, and then it can tell you what concentration and if cocaine is present or not. Alrighty, and then we have DNA based biosensors staying there. So, what we can do is we can use electrodes, we can use chips, we can use crystals to diagnose for clinical diagnosis and to check genome mutation. Alrighty, next we have a very upcoming arena for wearable biosensors. So, what wearable biosensors do is that they again use bioreceptors, and because we are wearing them, a very obvious lead should be that we want to detect what is happening inside the human body. That is the only reason why we are wearing them. For example, this ring sensor is designed in a way to check your blood pressure, to check the behavior of your heart. And because you can wear it, it is very comfortable for the patient to wear. So, what it has is it has your transmitter, RF transmitters, and then this is your finger, there is inner ring, there is outer ring, and then there is a photo detector here. So, every time the blood is pumped in the body, what happens is that there are pressure waves that are created. So, no pumping of the blood is relatively stationary, and then a pump comes, a thrust comes, and the blood moves. So, every time the heart pumps, there is a pressure wave created. The pressure wave hits the walls of our vein system, vascular system, which changes the pressure, which is what the systolic and diastolic pressure is about. Now, it also changes the flow of blood. So, what this optical business does is that it looks at when the flow increases or decreases, the optical property of your finger changes, and this is what is captured by the ring sensor. So, let us read more about wearable biosensor. It is a pulse oximetry sensor that allows one to continuously monitor heart rate and oxygen saturation in a totally unnoticeable way. The device is shaped like a ring, and thus it can be worn for long periods of time. The ring sensor is equipped with low power transceiver that accomplishes bidirectional communication with the base station and to upload data at any point in time. Next we have SMARTSHIRT from Georgia Tech. So, this is Georgia Tech wearable system, and the way SMARTSHIRT works is that the sensors have been woven into the shirt. This is non-stitch shirt by a very special weaving style, and then these sensors, they pick up your body signals. They pick up temperature, blood pressure, and what is going on in the body. So temperature, heart rate, and respiration rate. And if there is any casualty, if there is something not right, they will promptly give the information, which will be informed to the base station, and then once you have received it from a Bluetooth receiver, and then your doctors can tell you what to do. Now, what are the applications of SMARTSHIRT? Very, very important in combat. So, if you have a combat and let us say there is a toxic gas, and then the respiration rate would change, temperature of the body would change, the heart rate would change, and we want to get a quick information about how our soldiers are doing. We can either make them wear this, or when they come to us to hospitals or clinics, we can put this on their bodies and see. This will inform us what needs to be done to protect their life, to save their life. And it also can be used for chronic patients of heart diseases, respiratory illnesses, to get prompt feedback on if something is going wrong in their body, so that they can seek attention, seek help, and live longer. Alrighty, now let us look at biosensors on nanoscale. We were talking about nanobiosensors, so let us read more about them. So, molecular sheath around nanotube are developed that respond to a particular chemical and modulate the nanotube's optical properties. So, basically we can make nanotubes of some material, and then what we have is we make molecular sheaths around this tube. So, when it interacts with your analyte, its optical properties change, and then that can be transduced into a signal, electronic signal. Next possibilities is a layer of olfactory proteins on it. So, this is, by the way, your nose. It is a synthetic nose. So, what we have here is we have olfactory proteins which are specific to certain olfactory agents. Remember, olfactory means smelling. So, there are receptors in our body and in our nasal nerves which will actually pick up signals. So, that is how we can smell perfume. So, what we do is we take these proteins, olfactory proteins, on a nano electrode. We react with low concentration of odorants. Then doctor can use this to diagnose diseases at earlier stages. So, you must have heard about sniffer dogs, how can they smell diseases, how they can smell diseases really well. So, similarly, we can actually have a synthetic, or let us say an electronic biosensor based nose that can detect diseases early on, so that we do not have full blown cancer patients coming in for diagnosis and treatment. Right at the very start, we know already cancer is developing. Now, think about it, the same spot nose can be used for environmental contaminants. You just have to carry your nose, and it can smell pesticide, can smell metals. Next is nanosphere lithography, and what we have here is we use nanosphere lithography to make triangular Ag nanoparticles, silver nanoparticles, which are then used to detect streptavidin up to 1 picomolar concentration. That is a very low concentration. Then we have School of Biomedical Engineering, which has developed an antibody based piezoelectrical nanobiosensor for anthrax, HIV, and hepatitis detection. This is in US, a very big concern in US, anthrax for bioterrorism, HIV because there is no cure for it, and hepatitis. Alrighty, now where can you use these biosensors? You can use them, as we talked in the first lecture, for clinical diagnosis. You can use in food agriculture processes, for environmental monitoring, that is what we are interested in, and detecting warfare agents such as anthrax. So, this is multi, and in US, this is for US, by the way. There is a big market for biosensors, and we are noticing in India now our market is growing. So, in summary, the application of biosensors is for analyzing food, to study biomolecules and the interaction, for research purposes, to develop drug, to detect crime, to do medical diagnosis both in lab and clinics. This is where we are most interested in environmental field monitoring, quality control, industrial process control, detection systems for biological warfare agents, manufacturing of pharmaceuticals and replacement organs. And focusing on environmental applications, so the electrochemical aptasensors, these aptamer based sensors, they have been used to detect small toxin molecules such as algal toxins, pathogens such as Staphylococcus aureus, pesticides and heavy metals. So these are your biosensors, by the way, and here is a list from a paper published in 2014 of different analytes for which we have very good aptamer based sensors. So for theophylline we have RNA based biosensor, for cocaine again aptamer based biosensor, for chloramphenicol again RNA based DNA based aptamers, for tetracycline again antibiotics we have both RNA based, electrochemical based and aptamer based biosensor, and for mycotoxins we have fungal toxins, we have DNA aptamer based sensors. For endotoxins we have electrochemical based aptasensor. For bisphenol A, acetamiprid and pesticides we have DNA based aptamer sensors. For virus infected cells we use aptamers. For E. coli we have aptamer functionalized carbon nanotubes. So remember nanotube based sensors that we learned here. And then for anthrax we have nano aptasensor. So this is giving you an idea of the environmental contaminants, whether they are antibiotics, whether they are toxins, whether they are pathogens such as virus, such as E. coli, anthrax, or drugs such as cocaine that we do not want in our water, do not want in our food. So we have electrochemical aptasensors for them. These are all biosensors, and the scope is just continuing to increase because most of these sensors are very cost effective and robust, and they meet all our requirements. If they can be made in such a way that they are highly sensitive, very specific and very reliable. So alright dear students, this is all for today, and this is all for biosensors. In the next class we will talk about bioinformatics, and that would be the last and also very important part of our course. Thank you very much.